Hi, my name is Brianna Vargas Correa, and this is my Group Advanced Concepts Presentation 5 for Biochemistry 1. Once again, please excuse and please don't mind my voice, as I always lose it throughout the weekends. As I mentioned before, my involvement in the Portuguese community regarding musical theater and my performances are always on the weekends, and therefore I always strain my voice. For this part, I wanted to give more background information regarding cellular respiration. Therefore, cellular respiration is a process that allows your body to harvest a huge amount of energy from a single glucose. In fact, it's so efficient that you get 30 ATP for every 1 glucose molecule you eat. This exercise is going to break down exactly where each ATP comes from. This slide shows the generation of ATP. ATP is generated by the phosphorylation of ADP. As you see, ADP creates energy and therefore creates ATP. I will discuss more about the process as well as ATP and the other molecules. Overall, ATP is a complex organic chemical that provides energy to drive many processes in living cells, including muscle contractions, nerve impulse, and chemical synthesis. It is found in all forms of life. ATP is often referred to as the molecular unit of currency of intracellular energy transfers. There are a couple ways in which ATP is generated. One will be glycolysis. Glycolysis is one method of producing ATP and occurs in almost all cells. This process is an aerobic catabolism of glucose that converts a molecule of glucose into 2 molecules of pyruvic acid and 2 molecules of ATP. These molecules are then used as energy by various systems in the body. In eukaryotic organisms or organisms with a membrane-bound nucleus, glycolysis occurs in the cytosol. Oxidative phosphorylation also produces ATP and is a major producer of ATP in organisms. 26 out of 30 molecules of ATP generated from glucose are produced through oxidative phosphorylation. In oxidative phosphorylation, ATP is produced when electrons flow from chemicals known as NADH or FADH to oxygen. Aerobic respiration is another way that ATP is formed. Aerobic respiration also uses glucose to produce ATP. And as the name indicates, oxygen must be present for the process to occur. Without oxygen, aerobic respiration converts to anaerobic respiration, which only produces 2 ATP, compared to aerobic respirations of 34. Anaerobic respiration results in lactate buildup in animals, or alcohol and carbon dioxide buildup in yeast and plants. Similarly, FADH2 and NADH are created from FAD and NAD+ through reduction-oxidation reactions in the Krebs cycle during respiration. The cycle gives off small amounts of energy in the form of ATP and produces these compounds, FADH2 and NADH. In general, the citric acid cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate derived from carbohydrates, fats, and proteins into carbon dioxide. Also, the Krebs cycle is a series of enzymatic reactions that occur in all aerobic organisms. It involves the oxidation metabolism of units and serves as the main source of cellular energy. The first step is a condensation step, combining the 2 carbon acetyl groups from acetyl-CoA with a 4-carbon molecule to form a 6-carbon molecule of citrate. CoA is bound and diffuses away to eventually combine with another acetyl group. Citrate then loses 1 water molecule and gains another as citrate is converted into its isomer, isocitrate. Isocitrate is then oxidized, producing a 5-carbon molecule, together with a molecule of CO2 and 2 electrons, which reduce to NAD+ to NADH. This step is also regulated by negative feedback from ATP and NADH and by a positive effect of ADP. Steps are both oxidation steps, by which it releases electrons that reduce NAD+ to NADH and release carboxyl groups that form CO2 molecules. And ketoglucarate is a product of step 3, and a succinylate group is a product of step 4. CoA binds to this group to form succinylate CoA. The enzyme that catalyzes step 4 is regulated by the feedback inhibition of ATP, CoA, and NADH. A phosphate group is substituted for coenzyme A, and a high energy bond is formed. The energy is used in substrate-level phosphorylation to form either GTP or ATP, in this case ATP. There are 2 forms of the enzyme for this step depending upon the type of animal tissue in which they are found. One form is found in tissues that use large amounts of ATP, such as the heart and skeletal muscle. This form produces ATP. The second form of the enzyme is found in tissues that have a high number of metabolic pathways, such as the liver. This forms and produces GTP. Ultimately, in step 6, this is a dehydration process that converts succinate into fumarate. Two hydrogen atoms are transferred to FAD, producing FADH. The energy contained in the electrons of these atoms is insufficient to reduce to NAD+, but adequate to reduce FAD. Unlike NADH, this carrier remains attached to the enzyme and transfers the electrons to the electron transport chain directly. This process is made possible by the localization of the enzyme catalyzing this step inside the inner membrane of the mitochondria.